I'm HCS class of 2007, so this is my 5th year back in the program. It's been great. The past couple of summers I've worked with Dr. Patricia. I have a hard time pronouncing her last name. We worked in the cell signaling laboratory at the New York Health Center, and my project title is Random Cell Motility Analysis in API-1 Knockout Mouse Embryonic Fibroblast, which I'll explain. So what is cell motility? Cell motility is a term that refers to the movement of cells in the environment and movement of organelles within the cell. So any type of cell movement or the organelles within the cell is related to cell motility. And that's in order to fulfill a cell's role. So if a cell is seeking food, or if a cell is, like as the last presenter, like white blood cells, their goal or their function, and where they need to go, the cells, that has to do with cell motility. And that process requires energy. So the energy that's being used in cells is ATP, which fuels all cell movements along with the action of proteins. So you need ATP, the energy, and proteins to convert the ATP's energies in order for the cell to move. Unfortunately, the same process is critical when it comes to cancer. When cells become uncontrolled, when they divide uncontrollably, or when they move from place to place uncontrollably, that's when cancer can become a problem. How is cell motility measured? There are different methods out there, but the process that we used in the lab was the most difficult process, which was manual processing. So we actually had to trace cells manually over a long period of time, and I'll explain how we did it in a bit. We used this Zeiss Meta 510 confocal microscope to visualize the cell movement, and we used ImageJ software and Excel to do all the data analysis. Here I have a picture of the cytoskeleton. The cytoskeleton is the organelle, or the part of the cell that is responsible for cell motility or cell movement. It's made up of 3 parts: the microtubules, the intermediate filaments, and the actin network. The actin network is also known as the microfilaments, and that is the part of the cytoskeleton which we focused on, because as you can tell from the name actin network, it is made up of a globular protein, which is actin, and the ABI1 protein, which we are analyzing, is one of the proteins that helps make this actin, and it's found in the actin network, or the microfilaments. So the proteins, this is a list of the proteins that control the actin polymerization, depolymerization. Should I explain what polymerization is? Okay, so very simply, you guys know monomers and polymers? Okay, so a polymer is a large molecule made up of subunits, which are called monomers. These are the proteins, or the monomers, the subunits, that make up the actin network, or the actin part of the cytoskeleton. And one of the proteins that was being used, or that we found, was the ABI1 protein, which we wanted to study the effect of. Here's a picture that I drew of a cell and this actin polymerization, depolymerization process. Here's the nucleus, this is the cell, this is the cytoplasm. Here you have these little circles are actin, the actin, and one of the ways in which the cytoskeleton helps or assists in cell motility is that it sort of creates these tracks for the cell to follow. So as you can see here, the actin molecule would line up on top of each other in which track the cells should move with the help of these proteins that are around it. and this is called the polymerization or the formation of the actin put together or aligning together, and the depolymerization, which is the canceling out of the actin within the cell. And the proteins being used, again here, the list, and the ABI1 protein, which I'll go into that right now. So our hypothesis was if we remove the ABI1 protein from the cell, from the actin network, it will cause an impairment of the actin polymerization, depolymerization process, and as a result, because this process is important in cell motility, it will further lead to it will change how the cell moves and may actually lead to a cancerous state. Our methodology. Okay, so within the API1 knockout cells, by knockout cells I mean the cells that the API1 protein were taken out from. 30 hours after plating or after taking the protein out, images were collected at 15-minute intervals for over 960 minutes. So it was a very long process, and for every 15 minutes, we would trace where the cell went using this software, ImageJ. We would use this software to manually trace the cell and where it was going, and we measured for motility parameters such as migration path, distance, rate, directional persistence. All of these things were analyzed from our data. So the three main things that we were looking for in our results were migration rates, migration distance, and directional persistence. Migration rates is pretty much the ratio of the total length of how far the cell traveled over the amount of time. So it's just distance over time. Migration distance was just the translocation of the cell. So if it started at point A and ended at point B, showing the next slide, it wasn't the actual path of the cell, but more of the actual distance that it moved. So if I, for example, if I ran across the room and came back halfway, my translocation would be from here halfway because it's just final minus initial. And directional persistence was calculated as the ratio of direct distance over the total length of the migration path. Here's a picture of what it actually looked like. This is an example of 3 cells that we are following. If you can see here, it's not a very clear picture, but these are more cells. But just for here, so for example, migration rate would be how this is starting point A and this is point B, where it ended, where the cell just stopped moving. So the migration rate would be how far it went and how long the time. The migration distance would be from A to B, just this distance, not going around. And the directional persistence would be the translocation over the actual length that it went. Our results were interesting. So for migration distance, the normal cells that had the ABI1 protein, for migration distance and rate were both much higher than the knockout cells that didn't have the ABI1 protein. However, for directional persistence, the ABI1 knockout cells had a much higher directional persistence than the normal cells that were completely fine. So this led to the conclusion that there was an impaired lower movement rates of the API1 deprived cells, so the knockout cells had a lower rate and lower directional distance, which showed that there was a stronger directional persistence, suggesting a role of API1 of the API1 protein in polarizing the cells. This fact right here was not known until this discovery, which I believe they're working on publishing right now, but it was not very well known that the function of the API1 protein. Overall, although it's not 100% as of now, but it suggests that the API1 protein plays an integral role in the control of the actin polymerization, depolymerization, which heavily influences cell motility. Here are my references, and I'd like to acknowledge my mentor, Dr. Patricia, Dr. Sat, the New York Blood Center, Harlem Children's Society, HCS staff, and my good friend Ron. Thank you.